Today we're going to go over topic 19, which is all about Gregor Mendel and his genetics. So, where does an organism get its unique characteristics? It gets them from its parents' genes, who get it from their parents' genes and their parents' genes, and so on and so forth. So, they're all passed down from one parental generation to the next. Every living thing, plant or animal, microbe or human being, all has a unique set of characteristics that are from its parent or parents. So, parent being specifically if it's an asexual reproduction, parents, sexual reproduction. So, that movement from parent to offspring is called heredity, and the study of heredity is known as genetics. So, Gregor Mendel was a Austrian monk, and he worked in the monastery garden, and that's where he would mix and mess with pea plants, and that changed biology forever. So he would work on ordinary garden peas. They were really small, easy to grow, and they would produce hundreds of offspring in a very short amount of time. So they would use this model system. Scientists use all these model systems because they're very convenient. So they happen very, very quickly. And you can make a lot of offspring and you can see a lot of different characteristics at one time. By using these peas, Mendel was able to carry out in just one or two growing seasons, he was able to carry out hundreds of experiments. So it would be impossible to do for humans because you have to wait until the person's able to get pregnant and then able to have offspring, and then for that offspring to grow, and then them able to get pregnant, and so on and so forth. It takes too long. So Mendel knew that the male part of each flower makes pollen, which contains the sperm. So the male parts are right there. And then that the female part will take that pollen and produce a new flower, a new bud. And in that female part, there was eggs. So during sexual reproduction, we know if male and female reproductive cells come together, it's known as fertilization, and that produces a new cell. Pea flowers normally self-pollinate, which means the pollen will come back into the female part from the same flower, and it'll be the exact same flower over and over again. So Mendel's garden had several stocks that were true breeding, meaning they were self-pollinating themselves, and they would produce offspring exactly the same. So these traits of each generation would be the same. So a trait is a specific characteristic of an individual, such as seed color, plant height, it could be hair color, it could be hair texture, it could be any of those things. And they can vary from person to person. Just looking around the classroom, you can see how everyone looks different. Everyone has different traits. So Mendel decided to cross. So what he did was he would cut off the male part or the female part of one flower, and then he would get a little paintbrush, and he would get the pollen from the one that he cut off the female part and only had the male part. He would get pollen from there, and he would put it into the female part of the one that he cut the male part off. And that's cross-pollinating. So he had to prevent the self-pollination, which is why he cut those parts off. So that cross-pollination allowed a plant to have two different parents instead of the same parent. Mendel studied 7 different traits of each of these pea plants and 2 contrasting characteristics, so like green versus yellow. And he crossed the plants with each of these 7 contrasting characteristics to see what would happen to their offspring. That cross between parents with different traits is called hybrids. So those that, like the self-pollinating ones, those are called true breeding. The ones that are crossing, that are different traits, are called hybrids. So here are the 7 different traits that he looked at. He looked at seed shape, he looked at seed color, seed coat, pod shape, pod color, flower position, and plant height. Now, if you look here, you're going to see P, not only Ps, but you see the P generation right here, that's the parental generation. And then the F1 generation is the filial generation, but that's just like the offspring of the parents. So for each of these traits, all the offspring have the characteristics of only one of their parents as shown in the table. So round came here, yellow, gray, smooth, green, axial, and tall. So we think that when he first did the first cross, you would think that the other one disappeared. We know that's not true. So from these results, Mendel drew 2 conclusions. His first one was our first understanding and our current understanding of inheritance. So an individual's characteristics are determined by factors that are passed on from one parent to to the next. So it came from any characteristic can't just spontaneously show up. It has to come from the parent. So scientists call the factors that are passed from parent to offspring genes. Each of these traits Mendel studied was controlled by one gene that occurred in two contrasting. So these gene variations produce different expressions. All these different forms are called alleles, not alleles or anything like that. Alleles. Alleles. So his second conclusion is the principle of dominance. So that's the whole idea of having a dominant allele versus a recessive allele. And that dominant allele will overpower that recessive allele. Doesn't mean the recessive allele disappears, it just means that it is not being expressed in that generation. So he learned that tall plants were dominant to short plants. And then the same thing for yellow seeds versus green seeds. So how are these different forms of gene distributed to offspring? So during gamete formation, the alleles for each one will segregate. So this is where meiosis came into play. So each gamete carries only one allele for each gene. So I will show you that when I go over how to do dihybrid crosses in a little bit. But the law of segregation, which is that whole idea of them segregating into one specific allele in each gamete, will be shown, like I said, later. But for right now, you guys are going to be looking at segregation and what happens here. So he wanted to find out what happens to the recessive alleles. So he allowed all 7 kinds of F1 hybrids to self-pollinate. So he wanted to see them pollinate themselves. The offspring of the F1 are called the F2. So these were now the parents, and they created this. So in Punnett Squares, we're going to use mathematical probability to help predict the genotype and the phenotype. Now, probability is just a percentage. One thing I'm going to explain to you guys now is when you have that whole square of Punnett squares, which I'm going to show you how to do in case you don't, when it has that 25%, 25%, 25%, 25%, that doesn't mean that 1/4 will always turn out that way. It means that there is a 25% chance that you will have that trait. So I always use a example of if I have 5 girls, one after the other, what is the likelihood of me having a 6th girl? Most people are always like, oh, it's 1/6 or 5 out of 6, whatever it is. And I say, no, it's always 50%. There's always a 50% chance of having a girl or a boy every single time, no matter how many boys or girls you have in a row. So Mendels Cross produced a mixture of tall and short plants. And so you see that here, you're going to see tall, and these are the alleles right here. So this is law of segregation right here, where one allele went to each one. So this is all sperm or egg, whichever one you want to decide to connect to it. But it's all segregated. And then they are going to form these as they cross, as they self-pollinated. So each one had one tall and one short allele, and then half of the gametes they produced would carry the short allele. So right here, it's heterozygous. So because the T allele is recessive, the only way to produce a short plant is for 2 gametes that carry the T allele to combine. And then we have 2 gametes, so the probability of both gametes carrying the T allele is half times half equals 1/4. So right here, that's why that shows up. So the predicted ratio is 3 dominant to 1 recessive. So that's when, if we did a Punnett square, you would see that it is 75% chance of it being dominant versus 25% chance of it being short. Now, organisms that have 2 identical alleles for a particular gene, so capital T's or lowercase t's, are called homozygous. So this is homozygous dominant is the capital T's, and then homozygous recessive is the lowercase t's. Now, if it has a capital T and a lowercase t, these are called heterozygous. So every organism has a genetic makeup as well as a set of characteristics. Now, if you're seeing the tall pea plants, you're seeing that physical trait being expressed, that is a phenotype. So the P, think physical phenotype. Now, you're not ever going to see their genotype because that's a genetic makeup. So unless you have the equipment for it, you're not going to see their actual genotype, but genes, genotype. So phenotype, physical, genotype, genetic. So there's 3 different types of genotypes. You have the heterozygous, the homozygous dominant, and homozygous recessive. And as it's inherited, the phenotype is formed, so it shows through, it's being expressed. 2 organisms may have the same phenotype, but different genotypes. So that being said, like the heterozygous and the homozygous dominant. So one of the best ways to predict the outcome is with a Punnett Square, which is what we're going to do now. Okay, so we're going to be doing a Punnett Square. And we're going to do 2 different types of Punnett Squares. One of them being a monohybrid. So this is a monohybrid, mono meaning 1. This is a monohybrid Punnett Square. And with a monohybrid Punnett Square, we're going to have 2 different alleles, the mom and the dad, or whatever parents. So we're going to have that, and we're going to have this. So we have 2 different alleles. Now we're going to segregate them for the Punnett Square. So we're segregating them by putting one B here, one B here, the other B here, another B here. Now, there is no specific requirement that allows or has the dominant one going first, but most people do that just to remember the fact that it is dominant over the recessive. So this one comes down, capital B, this one comes here, lowercase b, then this one's also going to go all the way here. We're going to have lowercase b, and this one comes down, lowercase b. Now this one's going to come all the way down here. This one's going to come here. Same thing, lowercase, lowercase. So without the mess of lines, it looks like this. Excuse my terrible handwriting. B, capital G. That's a little different. Now, with a dihybrid cross, it's going to be 2 different alleles. So let's say this is a tall yellow. So tall yellow, and then we have they are going to be with a short green. Now we have to segregate this. Now, like I said in the lecture, when you segregate, it's kind of like meiosis. So when we're segregating, we're going to be using the FOIL method here. So we're going to have the first ones. So it's capital B, capital G. So these 2. Then we're going to go with capital B and lowercase g. So then these outside ones. So this is the first one. Then we have capital B, lowercase g. Now we're going to go to lowercase b, capital G. And lowercase b and lowercase g. So that's 4 different alleles. Or 4 different gametes. This one's only going to have 2 different ones, but we're going to repeat it a bunch of times. Or not even 2 different ones. It's going to have the same one 4 different times. Now, when we make this dihybrid cross, it's a lot bigger. This happening. Obviously, I'm not a math person because I did not make this square large enough. All right. So we're going to put these here. So this is going to be big B, big G, big B, little g, little g, little b, big G, and little b, little g. And then we're going to put all those little BG here. Now, same way as the monohybrid cross, we're gonna keep it consistent, and we're gonna keep the like to like. So we're gonna go with the B's first. We're gonna have big B, little b, and then we're gonna have big G, little g. This came down, this came down here, this went here, and this went there. And we're gonna do that all the way through. So we're gonna have big B, little b, little g, little g, little b, g, g, hopefully you're picking up on all this. It's kind of staying the exact same way across everywhere. That's gonna just repeat over and over again here. I'm just gonna, this is all the same. Now, when we see how many times by probability, this is out of 16, so this is gonna show up, this one showed up 4 times. This one showed up 4 times. This one showed up 4 times. And then this one showed up 4 times. So that's going to be 4 out of 16, which is 1/4. All of these are 1/4 chance of popping up. So hopefully you understood my little lesson on Punnett squares. Sorry for all the clicking of the pen, but I can't control how loud that sounds on the screen. When you're segregating independently for the dihybrid crosses specifically, that's the principle of independent assortment. So that's where like one of the alleles will come down, and then the other trait, one of those alleles will come down as well. And then during the formation of the gametes, you get different traits throughout it. So I use the tall, short, green, or yellow. So one of those gametes will be tall and green, one of them will be tall and yellow, one of them will be short and green, one of them will be short and yellow. So you have quite a mix of different gametes for that. So Mendel wondered if the segregation of one pair of alleles would affect another pair. So he performed an experiment with 2 different genes as they passed from one generation to the other. That's a dihybrid cross, which I showed you guys in that video. And some of them look like this. This one's obviously a lot neater than mine, but that's what the dihybrid crosses are. So in a summary of all his principles, at the beginning of the 1900s, Thomas Hunt Morgan, he decided to use fruit flies as a model organism in his genetics experiment, unlike Mendel, who used the pea plants. So the fruit fly was an ideal organism because it could produce a lot of offspring, and it did really quickly in the laboratory. If any of you guys ever go to AP Bio, we will do a lot of genetic stuff with fruit flies. So before long, Morgan and other biologists had tested every single one of his principles, and they applied to not only pea plants, but also other organisms as well. So these can be used to study the inheritance of human traits and calculate the probability of certain traits appearing in the next generation. Now, there are some exceptions. There's always exceptions. Some alleles are neither dominant nor recessive. Many genes exist in several different forms, and they have multiple alleles, and many traits are produced by the interaction of several genes. We're going to show through here. So even though there are exceptions, Mendel's work was very important. In most organisms, genetics is really complicated. So majority of genes have more than 2 alleles. There's way more alleles attached to something. Your eye color alone is 16 different alleles. That's why there's so many different shades of eye color. Same thing with your hair, it's multiple alleles. Many important traits are controlled by more than one gene. So his principles alone can't predict traits controlled by multiple alleles or multiple genes. And we're going to talk about that. Now, this is one of the exceptions. We have a incomplete dominance. Incomplete means that neither dominant trait is expressed, can overpower the other. So these are both dominant. So we have red and we have white. So what ends up happening with incomplete dominance is it mixes. Another example would be a black bunny and a white bunny. When they have their offspring, the offspring would be gray. So this is incomplete dominance. Codominance is that, once again, you have two dominant traits, but instead of them mixing, they both show up. So black bunny, white bunny, you have a black and white bunny as the offspring. So like co-captains, both are showing up. This is multiple alleles, so instead of a single gene, this single gene can have multiple alleles. So blood type, there's a bunch of different blood types. Most of the population is O positive. I'm O positive. O negative is the universal donor. Most people think it's O positive, but it's actually O negative. Then AB positive can receive from everyone. But even these single lettered ones right here, they can be heterozygous for two different alleles. So these can be B and O, or A and O, because if the mother is O positive or O negative, and then the father is B, their children will be BO, which means they have the possibility to pass on both different blood, like one or both different blood types to their kids. So they can pass on the B or the O. Polygenic means many genes. Poly means many. So these are a wide range of phenotypes. Like I said, skin color and hair color and eye color are all ones that have like a lot of genes to represent all the different shades. Now, environmental conditions can affect your gene expression and influence genetically determined traits. So that environment plays a factor. If certain things are around you, if there's radiation around you, if you receive more sun, if like so many things can influence it. Doesn't always happen. So the characteristics of any organism are not determined solely by the genes that organism inherits. It can also be how it unfolds depending on the environment. A karyotype shows all the chromosomes grouped together in pairs, so the homologous chromosomes all together arranged in order of decreasing size. So what they usually use these karyotypes for is to determine whether somebody has a genetic disorder or not. So we are going to explore the human genome. That's the full set of genetic information that an organism carries in its DNA. A study of the genome starts with chromosomes, the bundles of DNA, and the proteins. We already know how they condense from mitosis, meiosis. Scientists figured out cut out of the chromosomes from the photographs and arranged them in a picture known as a karyotype. I'm going to show you guys a picture now. It shows the complete diploid set of chromosomes grouped together in pairs, like I said. So you have a normal human cell contains 46 chromosomes arranged in 23 pairs. 2 of the 46 chromosomes in the human genome are known as sex chromosomes. These determine an individual's sex. So females have 2 copies of X chromosomes, so they have XX. Males have XY. All right, so here we have human karyotypes right here. We have a female one and a male one. These are both normal. So you can see the pairs of homologous chromosomes here. Individual, like this is one chromosome, and this is the second one. These are all homologous. So here is what determines a male one. You have an X and a Y, and then in a female one, we have 2 X's. So this is normal. Now, this is somebody with a genetic disorder. So this is Down syndrome specifically, which is a trisomy, tri meaning 3, of chromosome 21. So you see 1, 2, 3. So this one has 3 chromosomes in 21, which determines that they have Down syndrome. Okay, so the X and Y chromosomes are obviously what determines those are sex. So those are sex chromosomes. The other 22 chromosomes, or 22 pairs of chromosomes, 44 human chromosomes, are known as autosomes. So those are what determines everything else. So transmission of human traits. Many human traits follow a simple pattern of dominance. The alleles for many human genes display codominant inheritance. Because X and Y chromosomes determine sex, the genes located on them show a pattern of inheritance, and these are called sex-linked. So color blindness is one of the traits that's sex-linked. So most males will be colorblind versus females. There are some cases of females having color blindness, but it has to be a trait that both parents are affected. So the genes located on X and Y show a pattern of inheritance called sex-linked. So the genes is located on the sex chromosome, color blindness being one of them. Now, we're going to be going over pedigrees, which is basically a family tree, but this family tree determines disorders. So males are square, females are circular. If it's shaded, they are affected. If it's not shaded, they're not affected. What it doesn't show on here is they will usually show carriers as having half of it. Carriers being that they have the trait with them, but they're not expressing it. So they're just carrying it. So it would show half the square or half the circle as a carrier. You don't usually get the diamond shape. If there is a line between a male and a female, they are married. If there is a line coming down from that married line, then they have a child, this one being a son. Here, you would see a son, a daughter, and twins. You don't usually get twin ones, but you'll see male, like something like that, where they have a daughter or son. So this is an autosomal dominant. Remember that autosomal means that it is not a sex-linked trait. So that means it's on the other chromosomes. A dominant condition is transmitted in unbroken descent. So each generation will have somebody who has the trait. And so in this case, the mother is either dominant, homozygous dominant, or heterozygous. The father is homozygous recessive. If we did a Punnett Square of this, you would be able to determine what the chance of the kids having it is, and as it's passed down. So obviously this daughter married somebody who didn't have the trait. None of their kids had it. This one, it ended up happening. With autosomal recessive, it barely pops up. So you would have barely any shades. Normally it's somebody's a carrier and you have to end up marrying somebody who is also a carrier for your kids to have it. That's like something right here. So it doesn't show up, and then all of a sudden it shows up. This is because this daughter is a carrier and this guy she married was a carrier as well. So in their kids, they ended up getting it. Now, these are a variety of different disorders that are autosomal. We have albinism, cystic fibrosis, galactosemia, PKU, Tay-Sachs, chondroplasia, Huntington's disease, hypercholesterolemia, and sickle cell disease. So all of these right here, the white ones are recessive. These blue ones are dominant, and this is a codominant allele. We will usually do things with cystic fibrosis where we talk about this, PKU, and sickle cell.